So to start with, I would like to thank the organizer for giving me the opportunity to present my research. And I also want to thank the ISITE to support this work. So I'm going to talk about the role of NLRP3 in Th17 cell biology. So to start with, I'm going to remember you that immune cells are very important to fight against virus and bacteria, but they are also very important to fight against cancer. When a cancer occurs at the beginning, the immune system can fight these cells and can limit the progression of the cancer. But at the end, the cancer, I would say, win the war and develop strategies to limit the immune system efficiency. Among the cells that can be involved in the immune system, you can find macrophages, monocytes, NK cells, B cells, and also T cells. And my researches have to, well, I work on T lymphocytes. You can divide T lymphocytes in two main families that are called CD8 lymphocytes and CD4 lymphocytes, and this project has to study CD4 lymphocytes. These lymphocytes can be divided in subtypes that are characterized by a protein that it expresses, and that is called, I would call them a driver. So this protein will induce the function of the cells, and also by the release of a specific cytokine that will allow a part of their function. In this project, I study Th17 cells that are characterized by the expression of our gamma T driver and by a large amount of I-17 production. Th17 cells have a very controversial role in cancer because you can observe two types of Th17 cells. On one hand, cells that are called regulatory Th17 cells and that are not able to control tumor growth. And I would say they help tumor growth because they express at the membrane level two enzymes that can allow to produce a molecule that is called adenosine, which has a very strong pro-tumor effect by reducing the effect of the other immune cells. During my presentation, I will call them Th17R. These cells do not produce interferon gamma, that is one of the main characteristics of the other one, Th17 cells, that are called effector Th17 cells, and that were described to have a real impact on tumor progression, and it seemed that this cell could induce tumor regression. During my presentation, I will call them Th17I. For this project, I work on a specific protein that is called NLRP3 that was initially described in macrophages as a protein that will allow to produce the molecules that are responsible for inflammation. But in this context and in these cells, in Th17 cells, it is a very different mechanism that occurs. And we tend to say that it's a non-canonical function of NLRP3. So what we did, we isolated from mice naive CD40 cells, so from mice that we call wild-type mice, that is mean that they are control mice without any genetic alteration. And also we performed the same experiment using NLRP3 deficient mice. So we isolated naive CD40 cells and differentiate them in vitro in Th17I or R. And then we perform a global transcriptome analysis using RNA-seq and compare the different cells. What we observe is that we can easily discriminate Th17R here in blue and Th17I in red. And when Th17R were deficient for NLRP3, they move from this group, this blue group, to the red group. That means they tend to look like Th17I. So to go further in this characterization, we wanted to analyze the specificity of this subtype. And first, we analyze the production of interferon gamma. And we observe that Th17I can produce a large amount of interferon gamma as it was described in the literature, and Th17R are not able to produce this cytokine. But when this cell, the blue one, were deficient for NLRP3, they can produce interferon gamma. And this is true at the gene expression level and also at the protein level if you see a global analysis or a single cell analysis. As I said before, there is another characteristic that can discriminate these cells, is the expression of ectonucleotides at the membrane. So first, we analyze the expression of both ectonucleotides, CD39 and CD73, and observe that Th17I do not express neither neither CD39 and CD73. On the other hand, the cells that are called Th17R can express both, as you can see here. But when you study the cells that were deficient for NLRP3, there is no changes in the expression of these molecules at cell surface, as you can see here on the graph. We also evaluated whether these cells are capable to produce adenosine, which is the small molecule that can induce immunosuppressive activity, and there is no major changes in the production of adenosine. So our cells deficient for NLRP3 gain some of the characteristics of Th17I, but not all the characteristics. So we wonder whether they were pro or they have pro or anti-tumor effects. To evaluate that, we perform what we call a cell therapy experiment in mice. To do that, we isolate cells from mice that are either controlled or deficient for NLRP3, and then we differentiate in vitro the cells into Th17. Then we injected these cells into mice bearing tumor at the lung level. So to evaluate the efficacy of the treatment, whether the cells are have a pro or anti-tumor effects, you isolate the lung of the mice and you count the tumor that are black dots on the lung. Here is the control with that treatment, and you can see that when you treat the mice with Th17I, there is a slight decrease in the number of lung tumor foci, and when you injected the mice Th17R, it increases the number of lung tumor foci, confirming that these cells are pro-tumor cells. When you injected the same cell but deficient for NLRP3, you not only lose the pro-tumor effect of the initial cells, but you also gain an anti-tumor effect more important than Th17I. To understand, to go further in this demonstration, we wanted to identify models, cancer models, in which Th17 cells are a very important effect. To do this, we injected different cancer cells in mice, and then we treated them with an inhibitor of Th17 cells that is called digoxin. As you can see here, for this model, MC38, that is a common model, there is no effect of the treatment of the digoxin treatment. But in this both model, B16F10, that is a melanoma, and LLC1, that is a lung tumor, you can see a decrease in tumor growth when the mice were treated with digoxin. So these data indicate that these two models are dependent on Th17 cells. To evaluate whether our strategy to change Th17 cells from bad one to a good one is efficient in this context, we developed mice for which NLRP3 is deficient only in CD40 cells. And we performed the same experiment. And what we obtained is that without NLRP3 in CD40 cells, there is no change in tumor growth for this model that is not dependent on Th17. But in the two other models, you can see a decrease in tumor growth that is tend to confirm that Th17 cells are targeted by the deficient of NLRP3 in CD40 cells. To understand how it works, we first propose that this cell gain cytotoxic capability. To verify this hypothesis, we perform a cold culture, so we mix together Th17 and cancer cell, and we evaluate whether this cell can kill cancer cell. And as you can see here, there is no difference between the two cells. So to go further, we reproduce the experiment in which we injected tumor cell in either control or in NLRP3 deficient mice and analyze the tumor infiltrate. And we observed first that there is a decrease in Treg cells, and these cells were described to have a very strong pro-tumor effect. And on the other hand, we also observed an increase in CD8 activity, and these cells were described to have a very important anti-tumor effect. To go further, we analyzed among the genes that were identified in the previous experiment, in the RNA-seq experiment, and we observed that specific cytokine that is called ISITE-34 is decreased on one hand, and that several cytokines that were described to increase CD8 activation are increased in NLRP3 deficient Th17 cells. So to summarize these data, I just want to remember you that there are two types of Th17 cells. On one hand, the Th17R that have a pro-tumor effect, and the Th17I that have an anti-tumor effect. When you deplete NLRP3 in the first one, they change and then become anti-tumor and have a real anti-tumor effect. And probably this occurs because there is a change in the tumor microenvironment from an immunosuppressive environment to an immunoeffector environment. So that leads to tumor regression. So to finish, I would like to thank all the people working with me on this project. Theo, who performed most of the experiment.
Maybe quickly, if you don't mind, because otherwise we won't have questions.
Everybody. Thank you for your attention.
So thank you so much, Frédérique. So as a chairman, I've got the privilege to have a question. So could you explain us how this inhibitor of yours is working to deplete the Th17? Is that so specific?
It depends on the dose you use. According to the dose, you can gain in specificity. The dose we used, we know that it targets only the transcription factor Rho gamma T, which induce the depletion of Th17 cells.
Would that be available for clinical trials? Is it toxic in vivo or not?
I don't remember. In human, I don't remember. In mice, it's always the same. According to the dose, it can be more or less toxic. And the dose is quite, it's not very important to kill specifically Th17 cell.